Hi everyone, this is Kizaya Youngwest and I'll be discussing a parasite about sarcocystosis. We have our contents for this topic. We have the description, distribution, habitat, morphology, life cycle, pathogenesis and clinical features, immunity, diagnosis, treatment, and lastly prevention and control. So what is sarcocystosis? Sarcocystosis are intracellular protozoan parasites. When we say intracellular, it means within a cell. Then a parasite with an intermediate definitive host life cycle based on a prey-predator relationship. However, majority infects the mammals and some infecting reptiles and birds. Also, there are more than 200 species of Sarcocystosis, but only 26 species have known life cycle. And did you all know that Sarcocystosis was first found in a mouse by Mister in 1843? And now for the distribution, so it focused on animal sheep and goats, which is the common intermediate host and definitive host. This is a compiled data from 14 countries worldwide and this based on study. So the highest infection rate in sheep was found in Italy, Lithuania, and Thailand, and the lowest infection rate was found in Iran. They are in the higher risk because they into farming. And also, the highest infection rate for goats was reported in Malaysia and the lowest was found in South Korea. Infection rates were associated with the sampling site and detection method. So next is the habitat. So sarcocystosis were found in intestinal and cardiac muscles. Now let's move on to morphology. So this is the structure of sarcocyst. Its shape is oval and have a transparent appearance and has two sporocysts which tend to 18 long. And inside the sporocysts have four sausage-shaped sporozoites. And the oocyst cell wall is clear. However, if the sarcocyst will be out from the body or when it pass the stool, the sarcocyst cell wall would dissolve after pass out the body, and the sporozoites cemented together and can infect already. Now let's proceed to sarcocystosis life cycle. So the sporocyst and thin wall oocyst pass in feces. So the parasite will get in the feces, also in the contaminated food and water. After that, the sporocyst ingested by cows, pigs, sheep, goat, and so on. Then the sporocyst will rupture and release sporozoites. So sporozoites is a cell form that infects a new host that enter endothelial cells of blood vessels and undergo schizogony. Schizogony is asexual reproduction by multiple segmentation. These zones rupture, releasing merozoites. Merozoites is capable of initiating a new sexual or asexual cycle of development. Then merozoites penetrate muscle cells and develop into cysts with bradyzoids, then cysts with bradyzoids ingested in undercooked meat. So, in the internal bradyzoids release from rupture cysts, then will enter intestinal cells. After that will undergo fertilization and immature cysts exist host. Additionally, this life cycle shows to you that humans for some reason directly ingest oocysts or sporocysts containing sporozoites, it causes muscular form. On the other hand, when human ingests sarcocysts containing bradyzoites, it causes intestinal. Next is the pathogenesis and the clinical features. It is asymptomatic and clears spontaneously even without specific treatment. Nevertheless, if asymptomatic, the infection is called sarcosporidiosis or sarcocystosis, which has two forms. First is the intestinal form, ingestion of sarcocysts containing bradyzoites. So occasionally, mild fever, diarrhea, chills, vomiting, and respiratory problems may occur. Second is the muscular form, ingestion of oocysts or sporocysts containing sporozoites. Symptoms such as myalgia, muscle weakness, and transitory edema may occur. In these cases, humans are dead and intermediate hosts. Next is the immunity. There isn't a general immunity across all sarcocystic species, as each may elicit a different immune response. It's essential to maintain overall health to support the immune system's ability to combat such infections. Sarcocystosis is a parasitic infection caused by ingesting raw or undercooked meat containing the sarcocyst parasite. There isn't a specific immunity for sarcocystosis, but cooking meat thoroughly can prevent infection. Next is the diagnosis. To diagnose this type of parasite, this needs stool examination, a direct fecal smear, and a fecal flotation. Next is a biopsy of infected muscle stained with H&E. Next is the phylogenetic analysis. This is analysis of morphological and molecular data. And to diagnose intramuscular infection, we're going to base on its symptoms. And for treatment, currently there is no proven treatment for either intestinal or muscular sarcocystosis, but we can prevent the inflammation or the symptoms of the infection. We can use albendazole, prednisolone, metronidazole, and contrimazole. Lastly, the prevention and control. We can prevent sarcocystosis by cooking the meat thoroughly with 160 Fahrenheit or 71 degrees Celsius, especially the pork and beef. Use a meat thermometer to determine that the meat has reached a safe minimum internal temperature. Additionally, human consumption should not be exposed to contaminated water and feed containing. However, use toilets or other sanitation systems like chemical toilets to dispose of feces. Then especially, we are going to wash our hands, then drink and use safe water, then clean the food preparation areas.